As we continue our study of biotechnology, we're going to talk today about DNA profiling and genomics. So DNA profiling is when you analyze the DNA fragments, the pieces of DNA, to figure out whether they come from the same individual. We're going to use restriction enzymes to cut the DNA into pieces, and we're looking mostly at the non-coding parts of DNA and comparing those with the same pieces of somebody else's DNA. So what this does is it compares genetic markers from those regions that shows differences between individual people, and we're going to have to enlarge the sample. We're going to have to amplify or copy the markers so that we have enough to look at to compare. So first thing we do, let's say we have some blood from a crime scene. We're going to find the markers that we're looking for. We're going to amplify those by a method called PCR, which we'll talk about in just a minute. And that will be our sample in the first section here. And then we have two suspects. We're going to do the same thing to their specimens. And when we put this through an electrophoresis gel, electrophoresis is a process that uses electricity to separate the DNA pieces into dips by size. The smallest pieces go farthest and the longest pieces are closest to the source. Then we can see that there's a pattern that matches here between suspect 2 and the crime scene. So what that tells us is that suspect 2 is the person who left this evidence here. It does not tell us that he's guilty or that he's innocent or whatever. It just says that this DNA in this sample came from this suspect, because it wouldn't be identical otherwise. So we're going to use a method called PCR, which stands for polymerase chain reaction, to amplify or enlarge a segment of DNA molecule. PCR is kind of like a copy machine for DNA. And so we're going to use a couple of different kinds of chemicals with it. We're going to have some primers that are short single-stranded DNA molecules that are complementary to the ends of the target sequence that we're looking for. And then we're going to run it through this cycle, this three-step cycle, that every turn of the cycle, every time you run it, it doubles the amount of DNA. So let's look how it works. So we're going to start here with our original piece of DNA, okay, and we're going to use heat to separate the DNA strands. Remember that the two parts of the double helix are held together by hydrogen bonds, and heat breaks down hydrogen bonds, so that allows the strands to separate. And so we're going to add the primers to our solution, and the primers are going to bond with the ends of the target sequence. So here we have a primer bonded with the 3' end of this strand and also with the 3' end of this strand. And then we're going to add DNA polymerase, the enzyme, and free nucleotides, and DNA polymerase is going to do its job and make a copy there of the DNA. So now we've got the first cycle, we have from one molecule we have 2. In the second cycle, our 2 molecules that we started with are going to become 4. In the third cycle, they're going to become 8, and so forth. So every time you run the cycle, you get a doubling of the pieces of DNA that you have. It doesn't change them in any way, it just increases the number of them because it's copying those segments one after the other and after the other. So in a matter of several hours, you can get thousands of copies, and that increases the size of your specimen greatly, which is very helpful if you have a very small specimen to begin with. So again, we can amplify the DNA from a small sample. We can get results fairly rapidly, and then we've got this reaction that's copying only the target sequence that we're looking for and not the other thing. So it's very, very sensitive to what we're looking at. Then we're going to take these samples and we're going to put them through the process of gel electrophoresis. This is going to separate DNA molecules based on size. So here's how it works. The samples are placed in an opening at an end of a gel. The gel is porous and allows molecules to pass through. We're going to apply an electrical current to it. The DNA molecules are going to move from the negative electrode to the positive one because DNA itself has a negative charge, overall negative charge, and it's going to be repelled by the negative, attracted to the positive. You know, electricity flows from negative to positive because electricity is a flow of electrons, which are negative charges. So as the electricity is pushing these pieces of DNA through the gel, the shorter DNA fragments are going to move through the gel faster, and they're going to travel farther through the gel than the ones that are larger. And so what we'll see at the end is that we see the DNA fragments appear as bands. Now, in most cases, you're going to have to stain or use some kind of radioactivity or fluorescence in order to see the bands, but they're there. And then the bands are the collection of the DNA molecules of the same length. So their position on the gel tells us how long they are, and the thickness of the band is going to tell us that we've got more or fewer of the bands at that particular size. So here's just a picture from your book that kind of summarizes what we just said. Here's the gel, and it's got these opening wells in the end. We're going to put our samples in here. Again, we're going to run electricity, negative to positive. That's going to push the pieces through the gel. And again, the smallest ones are going to go farthest. The largest ones are going to stay behind. And then when you're finished, then you'll see a pattern here. And again, you can look at the pattern and see what matches what. This one looks like it might be determining that these two parents produce a child with this genetic profile. And a lot of times, here's a picture of a completed gel under fluorescence to show you where those bands are. You have to put some kind of a dye or kind of a marker on the DNA strands so that they'll show up. Now we're going to use this kind of analysis to as a DNA profile, and a profile can be used for a lot of different things. It can be used in a crime to determine guilt or innocence. It can be used to settle questions of paternity if you're not sure who the parents of this child are that truly are, then they can use DNA analysis to show that. Sometimes they're victims of accidents that are unrecognizable, and so you can use it to identify victims of accidents. And you can also use it to probe other things that are not involving humans. We can use it in determining genetic evolutionary relationships. There are lots of different things it can be used for to show how closely or how distantly related different animals or groups of animals are. So this process is used to analyze this is called RFLP, which is restriction fragment length polymorphism. So we're going to start with, we have first of all a single nucleotide polymorphism. That's a variation at a single base pair within the genome. And this is because of substitution. Remember, you can substitute a lot of times one base for another and still end up with the same amino acid in the sequence because there are multiple codons for each amino acid. So the RFLP is a change in the length of the restriction fragments because of the single nucleotide polymorphism that alters the restriction site. If the restriction site is looking for GAATTC and you change one of those T's to an A and an A to a T, then the sequence is going to be different, and that's going to switch where the piece is going to be cut. So the RFLP is a very handy thing to differentiate some of these from each other. It involves making the fragments, of course, with the restriction enzymes, and then we're going to sort them out again by gel electrophoresis. So here we have our restriction enzymes added to our samples. We've got the two different samples of DNA. We've had a change here at this first base pair. We've got an A and a T instead of a C and a G, and that's going to change the pieces that are cut. So this piece of DNA would be cut into 3 pieces. We have 3 bands there. This piece, since this is changed, it doesn't the restriction enzyme we're using does not recognize that sequence as a place to cut, so it goes on down to the next one. So we'll only have 2 bands because we have 2 pieces of DNA from that piece right there. Now, when you look at a DNA profile, what you're looking at is comparing the bands. And so let's just look at a couple of samples. Here we have a lot of times you'll have on a gel you'll have a standard, which is kind of like a ruler. It tells you basically how big these pieces are, how far they have traveled, and so it helps us when we're comparing the profiles to each other. So here we have 2 parents, a man and a mother, and we have a child. And what we're trying to see is if these 2 parents are the parents of the child. Maybe we know that this is the mother, but we don't know whether this is the father. Well, the child is going to have half of its DNA from its mother, the other half is his father. So what that means is the pieces of DNA, when you cut the DNA with the restriction enzymes, the bands that form in the gel are going to be either the same as one from the mother or one from the father. So here, this band right here is the same as one from the father. This band here, mother, mother, father, mother, father. Well, every band that we see in the child's profile matches up with one from either the mother or the father, so it is very likely in this case that this is the father of the child. In a crime scene, you're going to be comparing suspect DNA to DNA that was collected at the crime scene. So here's my crime scene DNA, and what you're going to look for is a suspect whose profile matches the crime scene. So again, they may have some bands in common, like you see through every now and then here, but the one that's going to match up exactly is going to be the one that was at the crime scene. So we have here suspect 2 that have bands that match up in every case to the bands that you find on the crime scene DNA profile. Now, and one more thing that we can do with genetic engineering and biotechnology is the study of genomics. This is what's been going on in the last few years when they've studied the human genome and genomes of many other organisms around Earth. It is the study of the organism's complete set of genes and their interactions with each other. Now, the first genomic studies, of course, were focused on prokaryotes. Again, they're smaller, they have smaller genomes, it's much easier to figure it out. And then since that time, with the prokaryotes were first done, many eukaryotic genomes have been investigated. There are genomic studies of all different kinds of animals, plants, fungi, and so forth. What we've seen in recent years is the completion of the Human Genome Project and comparing the human genome to genomes of chimpanzees and gorillas and all other kinds of animals. And so you can see the genetic relationships and the evolutionary relationships. What we see from genomic studies is a 96% similarity between chimpanzees and humans. In other words, if you compare the complete genome of a chimpanzee with that of a human, they will be 96% the same and only 4% different. The differences are going to be in various areas. Obviously, there's going to involve speech and reasoning and things like that, but it's very interesting to look for where those differences occur and what chromosomes they fall on and so forth. So evolutionary relationships is one way this is really used a great deal. We have discovered the functions of some of these disease-causing genes in humans because of being able to compare them with similar genes in various other organisms, such as yeast. So lots of interesting things have been discovered and will continue to be discovered about the secrets of the genome when we're able to compare them to other species.